Thank you, Rodney, for that introduction, and thanks everyone for coming. Today I'm going to tell you about how I use population genetics to understand the spread of two invasive vines on a small scale in urban centers in the southeastern United States. And I'll tell you what I've learned about how to best manage them. Before I get into that, I want to read an excerpt from a poem by James Dickey, an American poet who lived from 1923 to 1997. Japan invades. Far eastern vines run from the clay banks they are supposed to keep from eroding, up telephone poles which rear half out of leafage as though they would shriek, like things smothered by their own green, mindless, unkillable ghosts. In Georgia, the legend says that you must close your windows at night to keep it out of the house. Invasive species are organisms that have been transported purposely or accidentally to a different area, and they are harmful in their new environment. You probably know at least some of the invasive species I'm showing here: aquatics like Asian carp, zebra mussel and lionfish, insects like emerald ash borer and Asian longhorn beetle, vertebrates like python and wild boar, and plants like kudzu and bamboo. A species must go through several steps before it's classified as invasive. It must be transported from its native range to an introduced range. It must establish a breeding population without the help of humans. It must spread from the initial site of introduction, and it must have some impact. And this process can take a long time with lag phases before establishment and spread. And there's a lot that we don't understand about the spread of invasive species. For example, the rate of spread, the distance of spread, and the pathways of spread. And this is what I'm interested in. And I'm also interested in how we can decrease the impact of invasive species. Urban areas are heavily modified habitats characterized by high human density and lots of impervious surfaces. They have altered biogeochemical cycles and increased deforestation and pollution. And urban areas create environments that allow invasive species to thrive. Species that invade often prefer deforested areas or warmer habitats that are facilitated by the heat island surrounding cities. And they're often introduced without their enemies. This is a view from Google Earth of Staten Island looking into Brooklyn. And I'm showing this picture because at this scale you can see just how modified the habitat is. And you can also see a city block covered in kudzu. So the big questions I'm interested in are: how do invasive plants spread, and how can we use information about their spread to prevent their spread? To get at these questions, I studied wisteria. Wisteria is native to Japan and invasive in the eastern half of the United States. It was introduced in the mid-1800s as an ornamental plant, and it continues to be planted in residential gardens across the country. It reproduces sexually and asexually, and here we see lots of purple wisteria flowers behind this house. I also studied kudzu. Kudzu is also native to China and Japan and invasive in the eastern half of the United States. It was first introduced in 1876, and then in the early 1900s, it was imported, cultivated, and distributed on a wide scale by the Soil Conservation Services to prevent erosion. And it also reproduces sexually and asexually. And it is quite easy to find a picture of kudzu covering a house on the internet. These are pictures of kudzu populations. And what I want you to notice is how they grow in patchy distributions with distinct edges. There are lots of tubers growing underground, and so there are lots of physiologically independent individuals in these populations, but we can't distinguish them just by looking at it. Wisteria can similarly grow in patchy distributions, but it can also grow as a single individual that we can distinguish if it's planted in a residential garden. And so I'm using these plants to answer the questions: what are the dynamics of kudzu populations? What landscape features facilitate the spread of kudzu and wisteria in urban areas? And can we use mathematical models to provide science-based recommendations for the management of invasive species? Karen Bentley, a previous graduate student in the Mauricio lab, studied the genetic structure of kudzu across its invasive range in the United States. She genotyped 20 samples from 87 populations And here I'm showing a pie chart at each of the sampling sites. And the pie chart is showing the percent membership to each of 6 clusters that were distinguished by discriminant analysis of principal components. So what she found is that there is high diversity across this entire range, but there's no geographic pattern to the diversity. So we see all the colors everywhere across the entire range. So this is a static picture. We can learn about the distribution of diversity when these populations were sampled, but it doesn't tell us how the diversity is changing over time. And so I want to ask, what are the dynamics of kudzu populations? Specifically, do they persist over a long period of time? And is persistence equal across the range? So this is a distribution map, and where in counties where kudzu is present, it's colored green, and in counties where kudzu is absent, it's white. And there may be different things going on in different parts of the range. So here in the center may be different from the northern edge or the western edge of the range. So luckily for me, people tend to record the location of plants. In 1989, this guy, Edward Frankel, published all of the known sites of kudzu populations in and around New York City. And he published about 35 sites, and some of them had really specific location information. So for example, 22 Veranda Place, 5-minute walk from Borough Hall subway station. I know exactly where that is. So I can return to this site and see if a kudzu population still exists there. And in order to compare in a different part of the range, I went to herbaria to collect specimens with specific locations that I could know I was returning to the exact same spot and check to see if there's still a kudzu population there. So when I did this, I found that in the core of the range, 18 out of 19 populations persisted over an average of 34 years. And at the north of the range, 7 out of 21 populations persisted over 22 years. So populations can and do persist for a long period of time, and persistence is a lot higher in the core of the range. So the next thing I wanted to do was learn about the short-term dynamics of kudzu, what's going on at a much smaller time scale. So in order to do that, I had to return to a kudzu population much more frequently. And by repeatedly genotyping the same population, I can see if the genetic composition of populations changes over time. Kudzu is deciduous, and each November, the above-ground portions die, and it regrows in the spring, so it looks like this. It flowers in August, and seeds are mature in the fall, in like October. So what I did is I went to populations in the spring that looked like this, and I returned in the fall when it looked like this, and then again in the next spring. So I'm asking the specific questions: does diversity decrease over the summer, maybe due to competition for light resources, or does it increase due to seed recruitment? And then between the second and third sampling times, I asked, does diversity decrease over the winter, or does it increase due to seedling recruitment over this time? So at this point, no one knew how many genotypes were in each kudzu population, if it was one or more than one, and if it was the same over time or if it changed over time. So I picked 6 kudzu populations, and I sampled an average of 87 samples per population per time. So this actually was a really big sampling effort with almost 1,500 samples altogether from these 6 sites. And the reason why I sampled so many from each season and each population was to try to recover all of the diversity present in each population. So here I'm plotting rarefaction curves to see how well I did. And the numbers plotted are the slope. So what this tells me is I would need, on average, 42 more samples collected from a population to recover one additional genotype. So I really did a good job at sampling almost all of the diversity throughout the entire population. And so here are my results. So what I'm showing here is each circle is a clone, and if the clone was present at more than one time, I put the clone in the same column and drew a line through it. If the clone is only present at a single time, then it's only in season 1, season 2, or season 3, so it's only in this one row. So what this tells me is, are the number of clones the same over time? And what I find is that clones are commonly lost or gained. There are the same number of clones over time. The numbers are not statistically different between seasons. But it's not the exact same clones present because we have all of these lost and new clones in each population. So the diversity is not increasing or decreasing, but new clones are gained and lost. If clones are gained due to seedling recruitment, I would expect them to be at low frequency. So the next thing I tested was the frequency that I sampled all of these clones. So here I'm showing the frequency of clones where each clone is in a different color. And so the colors are only consistent within one population, but in order to not use like 100 colors, I repeated the colors. So blue is the same between these three, but it's a different blue as this. So the most striking thing to me is right here. Between season 2 and season 3, I have this huge turnover of about 80% of the samples in this population. And so this shows that it either may be from seedling recruitment, in which case a genotype can increase in frequency really quickly, it can grow really quickly, which is not that surprising for kudzu, or maybe it was a dormant tuber underground. And in other populations, there's up to 17% unique genotypes to a single season. So these populations really are a lot more dynamic than we would have expected, with new genotypes arising and potentially coming to a really high frequency in a short amount of time. However, the absolute levels of diversity are not increasing or decreasing. So at this point, I want to bring you back to my main questions of how do invasive plants spread, and how can we use information about their spread to prevent their spread. Specifically, I'm asking what landscape features facilitate the spread of kudzu and wisteria in urban areas. Population genetics studies often look at isolation by distance. We often care somewhat about how these things are structured in space. So here's a cartoon of a structure plot which shows very nice structure, a PCA which shows that the blue and red and yellow are really different. And then we can put this on a map. So we can say, are the blue populations closer to each other, and are the red populations closer to each other? But often population genetic studies don't use geographic information or use information about the landscape in a really substantial and rigorous way. But in order to answer my question about which landscape features are facilitating spread, I really need detailed information about the geography of these populations. Landscape genetics is a field that has been developed to provide this information and to bring together population genetics and landscape ecology. Landscape genetics quantifies the effect of landscape heterogeneity on genetic patterns to understand gene flow or adaptive genetic variation. Microevolutionary processes and landscape heterogeneity are quantified, and relationships between the two are explicitly tested. Because intervening landscape between populations is quantified, I have to use tools specific to landscape genetics to answer the question, what features in urban landscapes help invasive vines to spread? So here I'm showing a picture of kudzu growing along a railroad, on the banks of a river, and along a road. If you can see the road right down here. And I hypothesize that plants may be spreading along these features because they're often found along these features. I have a lot of the collections near them. In order to do a landscape genetics project, I need to collect genetic data, look for genetic structure, collect landscape data, and then test for a relationship between the landscape data and genetic structure. So now I'm going to walk through how I did each of these 4 steps. So in order to understand how vines are spreading, I have to collect samples from across the landscape. So I need a lot more populations than the 6 I had in the previous study. And since I'm interested in urban areas, I collected samples from across Athens and across Anderson, South Carolina. Anderson, South Carolina is about 50 miles away, and it has a roughly similar size, elevation, and climate as Athens. So here in Athens, I have lots of different localities. Because I tried to sample exhaustively from all of the kudzu populations in Athens. In Anderson, my sampling intensity was a lot lower, so I collected fewer samples, but I don't think there's less kudzu present in Anderson. Wisteria grows less patchily, so I have a lot more sampling sites. And again, I tried to sample from all of the wisteria that is present in Athens. But my sampling intensity was lower in Anderson, so I don't have as many there. I want to point out just how many samples I have. In this data set alone, it's almost 1,500. So I really have a lot of samples, and I need a genetic marker that I can, first, that can give me resolution to distinguish all of these samples. And second, a genetic marker that I can use and can afford to genotype all of these samples with. Sequence capture and dual digest RADseq are 2 common ways to make reduced representation libraries for next-generation sequencing. Sequence capture uses probes, which I'm showing in yellow, to target and enrich DNA sequences, which I'm showing in black. And because we need probes, sequence capture is expensive because the probes have to be designed and ordered, and we need prior genetic information to know the sequence of the probes. However, sequence capture controls the loci, and there's little missing data, so you really get a good data set for all this effort that you put in. On the other hand, dual digest RADseq sequences DNA fragments that occur between 2 restriction enzyme cut sites. It's cheap to do on a lot of samples, and there's no prior genetic information needed, but it doesn't control loci, and there's a significant amount of missing data. Because I have so many samples, I thought missing data would be an especially big problem, but I didn't have enough money to use sequence capture. So I set out to develop a new method of collecting genetic data. In collaboration with Travis Glenn, we developed RADcap, which uses elements of sequence capture and dual digest RADseq. And RADcap is cheap, no prior genetic information is needed. It controls the loci, there's little missing data, and it also makes this improvement that sequence capture and dual digest RADseq don't have. It allows me to remove PCR duplicates, and PCR duplicates can give a false confidence can artificially inflate read numbers and give you a false confidence in your genotype calls. So I want to tell you a little bit more about RADcap and how it makes these advantages and how well it works. RADcap is a further reduced representation library. I'm taking RADseq libraries, specifically a variant of dual digest RADseq, which I helped develop called 3RAD, and I'm performing a sequence capture on them. In this graph, I'm showing the number of loci out of all the loci that I designed probes for, how many I retain at different levels of sequencing coverage for a plate of samples. So here are 96 samples, and I'm recovering that many loci. The same plate with RADcap. I've gone up on the y-axis, so I've collected more loci, and I'm coming left on the x-axis, so I'm doing it with fewer reads. What this means is I'm reducing missing data, and I don't have to spend as much money on sequencing costs because I'm sequencing fewer reads. And this makes a huge difference when you have thousands of samples. Another way we can look at the amount of missing data in RADcap datasets is to look at the size of the data matrix when we allow different levels of missing data. At 0, I'm throwing away, so this is the percent of loci recovered and the percent of missing data permitted. With 0% missing data, I'm throwing away any locus that doesn't have a SNP call for any individual. At 50% missing data, I'm throwing away loci that have missing data for more than 50% of samples. So that's really a lot of missing data. And what I want you to notice is how flat these lines are from the 50 to 10 or 20% missing data. So it's only when I don't allow any missing data at all that I have this really sharp drop off here. And all of these numbers are non-zero. So the next thing I want to talk about is removing PCR duplicates. Here I'm plotting the percent of PCR duplicates that I sequenced in each of my 28 plates of samples. And lower is better because that means more sequences come from genomic DNA rather than PCR. In about half of my plates, I have less than 10% PCR duplicates, but there's this huge variance. And so the PCR duplicate identifiers that I've incorporated into the libraries are really important to have to identify and be able to remove PCR duplicates. Okay, so I've collected my genetic data and I've told you about this new method that I used to do it. I'm now going to look for genetic structure. To look for genetic structure, I use two types of eigenvector analysis. The first one is called discriminant analysis of principal components, and this is a non-spatial method. So using BIC, I selected the most supported number of clusters in each of my data sets. So here I'm showing one example from Anderson kudzu where 7 clusters has the lowest BIC values, and so that's the most supported number of clusters. What discriminant analysis of principal components does is it tries to maximize the differences between these groups of individuals, between these clusters of individuals. This method is non-spatial. It's not incorporating where these populations are, and gene flow can occur throughout the entire data set freely. So as much gene flow can occur between the two furthest sites as between the two closest sites. As a comparison, I used a spatial method called spatial principal component analysis. And what this does is it maximizes the amount of spatial genetic structure given a connection matrix. So I supply a connection matrix, and this is my hypothesis about how gene flow really works in this system. So here I'm showing one example, and I've picked Athens, Wisteria, and each of these dots is a sampling location. So if there's a line drawn between the dots, that means that individuals at that sampling location are neighbors, and gene flow can occur in one step between those two sites. If there's no line, like here, between these two at the very bottom, then gene flow cannot occur between those samples. So this is the non-spatial method, discriminant analysis of principal components. And what I'm showing here is, again, a pie chart of the percent membership to each of the clusters. So a lot of these sites belong to a single cluster, but some of them belong to more than one cluster. And I don't find any spatial genetic structure here. All the purple is across the entire landscape. All the colors are everywhere. So there's no pattern into where these clusters appear. On the other hand, spatial principal component analysis, here I'm plotting the first 3 principal components on the RGB color scale. So more similar colors means the samples are more similar genetically, and more different colors means the samples are more different genetically. And here I absolutely am finding spatial genetic structure. All the green guys are right here, and all the purple guys are right here. Okay, so let's move on to Athens, Wisteria. In Athens, Wisteria, I'm finding the same thing. There's no spatial structure. Here, all of the colors are all mixed up, whereas here there is spatial structure. Similarly, in Anderson kudzu, there's no structure here, and there is here. And in Anderson Wisteria, there's no structure here, and there is structure here. And so why am I finding this difference? I think that this spatial method does a better job at picking out the spatial genetic structure than this non-spatial method. By allowing gene flow to occur freely across the entire system, I'm saying that I expect spatial patterns to follow isolation by distance. The closer you are, the more similar you are. But I don't actually think that. I think that gene flow occurs differently in different landscape types. So it will be different through a forest versus along a road. And so a connection matrix is a better hypothesis than straight isolation by distance. And by using spatial principal component analysis, I can pick out these more fine-scale patterns. Okay, so now that I've looked for genetic structure, the next thing I'm going to do is collect landscape data. And to do this, I went to the National Land Cover Database from 2011, which looks like this, where different landscape features are in different colors. And of course, I'm only using data from right down here. And I collected roads and rivers and railroads from Tiger line data. I then consolidated the features into 8 different classes so that each of the features was present in more than 5% frequency on the landscape. So the first thing I noticed here is that Athens has a lot less of this green than Anderson does. And so I can quantify the amount of each land cover type across the landscape. And what I find is that Athens has a lot less pasture than Anderson, and Anderson has a lot less forest than Athens. Otherwise, these are pretty comparable. So I've collected my landscape data, and the next step is to test for relationships between the landscape data and the genetic structure. So here I'm showing a small matrix where different landscape features are in different colors. This matrix is actually a resistance surface, which is a spatial layer that assigns a value to each feature, representing the degree to which that feature impedes or facilitates connectivity of an organism. So we can think of this as a hypothesized relationship between the landscape variables and gene flow, and it's based on some underlying biology of the organism. So my goal is to add numbers, a specific cost, to these cells instead of colors. I performed a genetic algorithm in R to simultaneously optimize values for each land cover type in the resistance surface using least cost paths between kudzu or wisteria individuals within each city. So I seed the program with arbitrary numbers, and then it goes through many generations optimizing AIC with the resistance values as the independent variable and genetic distance as the dependent variable. So gene flow is only allowed along a single path with the smallest cumulative resistance, like this green path, and not allowed on any other path like this red one. So this program eventually gives me the numbers for each of these features. And low numbers facilitate the spread, and high numbers don't allow spread. So with the resistance values of each of these 8 feature types, I can assess the influence of different land cover types on genetic structure. I ran the optimization 3 times, and I normalized the values to a maximum of 1,000. So here I'm plotting the average of 3 runs. For Athens kudzu, the lowest resistance values are herbaceous, water, and roads, and so these are the features that facilitate the spread of Athens kudzu. This green dotted line is an arbitrary level where I've set to say what is a low resistance value and what is a higher resistance value. With these data, I have 2 questions. Which features facilitate spread, and does kudzu spread similarly in different cities? So what I'm showing here is that in Athens kudzu, herbaceous area, water, and roads facilitate spread, but in Anderson kudzu, water, lawn, and roads facilitate spread. And there are some big differences between the two. So developed is a lot lower in Anderson, and This means area with 20 to 50% impervious surface. I'm calling developed area. Herbaceous area is more inhibitory in Anderson kudzu than in Athens. Lawns facilitate spread only in Anderson, and forests inhibit spread more so in Anderson. I can do the same thing for wisteria. I can ask which features facilitate spread, and in Athens, it's water and roads. And in Anderson, it's water, lawn, and roads, and forests. And then ask, does wisteria spread similarly in different cities? And again, there are some big differences. So developed area is higher, has a higher resistance, which means it inhibits spread in Anderson. Lawn facilitates spread in Anderson, and forests facilitate spread in Anderson, but not in Athens. If I compare the spread between the different species in the same city, what I find is that in Athens, water, lawn, roads, and pasture, the two vines spread similarly along those, meaning water and roads are good for plant spread, and lawn and pasture are bad for both plant spread in Athens. On the other hand, in Anderson, both vines spread by water, lawn, and roads, and then neither plants spread by herbaceous area and pasture. So I've found that different types of plants so the same type of plant doesn't spread the same way in two different places, but different types of plants spread the same way in the same place. And I don't think this is reflecting the biology of these species. I think it actually has to do with the landscape of these the cities. So the landscape features may be correlated in the city. For example, you might have a gradient of really high impervious surface to a little bit less to a little bit less. And so there may be spatial autocorrelation in the landscape features. In addition, Athens and Anderson are two different cities with different development histories. And there may be a time lag between what's going on in the genetic data that I found versus how these landscape features have changed. So something can change from low development to high development, or from forest to road. So in summary, vines are spreading by roads and rivers, and in Athens they're also spreading by herbaceous area, and in Anderson they're also spreading through lawns, and wisteria is spreading through forests. So this indicates that we might be able to manage different plants the same way in the same city, but really this spread is dependent on where in the landscape these vines are. Okay, so now that we know these common features that plants are spreading across, maybe we want to target our management to along roads and rivers. So we can prioritize which patches to manage. But there are still a lot of patches along roads and rivers, and so it would be nice to have some more information to target management. So can we use mathematical models to provide science-based recommendations for management of invasive species? Some perennial invasive species, like kudzu, are difficult to remove because they store carbohydrates in their roots, and even after managing them, they can grow back. For species like this, is it better to manage a population that looks like this, which has never been managed, and last year it looked quite the same? Or is it better to go to a population that was managed recently, but still has some invader present at a low density? So I see these as two alternate strategies. We can either go to highly invaded patches or not low density patches. And so I developed a model to tell us which strategy would be beneficial to control these populations. So this is a city level model, and I'm thinking about, for example, all of the kudzu across the city, and I've divided the landscape into equal area habitats, and then I've classified, or equal area little patches, and I've classified these patches according to their invasion status. So we have empty patches, young patches, which are below the detection threshold and don't export propagules, mature patches, which export a lot of propagules, and controlled patches, which are recently controlled. So the difference between a mature patch and a controlled patch is controlled has a lower colonization rate, a lower maturation rate, and a lower cost of control. So in order to understand how well our model is working, I need to specify exactly what my goal is. So the goal I'm going to tell you about is, I'm going to call it eradication, and what it is the minimization of the number of mature and controlled patches across the landscape. So I'm not considering young because in this model they're undetectable. So here I'm plotting which alternate strategy, either controlling the highly invaded or lowly invaded patches, leads to a smaller invasion size after 20 years. And I'm doing this for all possible maturation rates and a wide variety of colonization rates. So treat mature will always be in pink, and retreat controlled will always be in blue. What I'm finding here is that unless you have a very low maturation rate, it's best to retreat controlled patches. If you have a very low maturation rate and a high colonization rate, it's better to treat mature patches. So this model can be used for invasive species with varying invader traits, like biological traits. So we can apply this model to species with a range of maturation and a range of colonization rates. So I have this little dot here, and I want to talk about this specific parameterization because I think that's what kudzu is. I think it takes a long time to mature, and then once it's mature, it colonizes new area very quickly. So for this specific kudzu-like invader, I'm now plotting the percent of the area occupied over time. So I'm not just looking at after 20 years, I'm looking across all of time. This black line is the amount of the area that would be full if we don't exert any control on any type of patch ever. The pink line is the amount of the size of the invasion if we only can treat mature, and then if we have money left over, we can retreat controlled patches, but first we're going to treat mature patches. And blue is the size of the invasion if we do the opposite, if we only retreat patches, and if we happen to have money left over, we can treat some mature patches. So what we see is that there's a trade-off. It really depends on how long you are controlling for, because if you're controlling for less than 15 years, it's better to retreat controlled, but for greater than 15 years, it's better to treat mature. The next thing I asked is, how does the available budget affect the optimal strategy? So here I'm plotting the size of the invasion by the number of patches that can be controlled in a year. And what I'm showing is for this kudzu-like invader, it depends, the optimal strategy depends on the budget that you have. So with a low budget, it's better to treat controlled. The size of the invasion will be smaller. But at higher budgets, it's better to treat mature. Then I asked, does it depend on the way the control action works? So I can imagine that different control actions work in different ways. One might kill the invader. It might have a really high lethality. Another one might decrease the propagules. So it might lower the colonization rate. And the last one might keep the patch in a controlled state for longer. It might decrease the maturation rate. When I compare these three different types of control, what I find is that by decreasing the number of propagules or slowing the maturation rate, you're not going to get rid of the invasion. It's decreased a little bit from if there was no control at all, but you're not being successful down here at 0. However, if you have a high lethality method, you can decrease the invasion size. And the strategy to use in this case is to retreat controlled patches. So I want to tell you about another goal of control. Maybe your kudzu invasion is just too big and you don't think that you can minimize the amount of mature and controlled patches, but you just want to go after these mature patches. So I want to minimize the ones that look like this, but I don't mind these. I don't really care how many of these there are on the landscape. So with this goal, I'm going to go through these graphs again, and what I find here is that for a much bigger area in parameter space, for many more different kinds of invaders, it's better to treat controlled patches than to retreat if your goal is beautification, which is what I'm calling this reducing the heavily invaded patches. If we look at the size of the invasion over time, we see that the treat mature strategy is always lower. It's always better to treat mature patches for this type of kudzu-like invader with a slow maturation rate and high colonization rate. And what about for varying levels of budget? We see again that it is better to treat mature no matter what your budget is. With the 3 different control strategies, by treating mature, it doesn't really matter how the control method works. All of them will decrease the size of the invasion. But if we look closely, this one decreases it first, so at around year 5. So if you have a choice, it would be best to use a control strategy that delays reversion, a control method that delays the reversion, and a strategy that treats mature. This model can be extended in a few ways. We can talk about the optimal strategy given varying budgets and control methods for different invader types. For example, musk thistle is invasive in the United States in about 40 states. It is native to Europe and invasive here in the southeast, including. And it grows really quickly, but seeds are not dispersed very far, so it has a low colonization rate. I can also incorporate control methods that work in different ways. So they can be really good at doing 2 of those things, not just 1. So what I've shown here is that optimal control depends on the budget and the time of control and the invader type and the goal. But the bottom line is that if your goal is eradication, it's more likely to be beneficial to retreat controlled patches. If your goal is beautification, it's more likely to be beneficial to treat mature patches. And for a quick colonizing invader like kudzu, it's more likely to be beneficial to treat mature patches. So in conclusion, I've shown that kudzu populations and clones are persistent over long periods of time. There may be lots of seedling recruitment and spread, but establishment of new clones is rare. And when it does happen, it may cause a really big turnover in populations. Spatial genetic structure does exist within kudzu and wisteria on a city scale. Both vines spread by roads and water, but the landscape influences how these plants spread. And for kudzu, it's best to go after unmanaged, heavily invaded populations, except under a few circumstances. Thank you for your attention, and I'm happy to answer any questions.